first off, we have María Mariner Faulí, who hails from Spain and is coming to us from Alvaro Rada Iglesias' lab. Maria did her undergraduate degree in biotechnology at the Polytechnic University of Valencia, and then moved to Cantabria to join the Rada Iglesias lab for her master's and eventually PhD in developmental genomics at IBTEC. And looking at Google Maps, I'm extremely jealous of the locale in which she's done her training. It looks much nicer than some of the cold places in North America that I've worked. But today she's going to talk to us about a really cool project that she's been working on involving the ZIC2 and ZIC3 transcription factors. And with that, I will let Maria take it away.
Thank you, Jackson. It is indeed beautiful. I completely agree. I love living here. Cantabria is awesome. So, thanks for the introduction, and thanks to the Fragile Nucleosome team for giving me the opportunity to present part of the project I developed during my PhD, and which is now publicly available as a preprint on bioRxives. So the title of my talk is Dual Regulatory Role of ZIC2 during in vitro neural induction. And as you would expect, the project focuses on ZIC2, a member of the zinc finger of the cerebellum family of transcription factors, which, among many other functions, plays a crucial role at different stages of brain development. And the functional relevance of this transcription factor is highlighted when examining its associated loss of function defect in humans, which is also recapitulated by mouse models. Holoprosencephaly. Holoprosencephaly is a congenital brain malformation resulting from the incomplete separation of the two brain hemispheres. And the classification of the disorder is based on the origin of the defect with respect to the dorsoventral brain axis. If the defect originates ventrally, we are dealing with the so-called classical HPE, while if the defect originates dorsally, we are dealing with the so-called middle interhemispheric. And what is really interesting about ZIC2 is that so far it's been the only gene associated to both types of HPE, which emphasizes the relevant role of ZIC2 at different stages of brain development and puts the focus on studying ZIC2 molecular function during the earlier stages of neural development, aiming to know more about the specific gene regulatory network controlled by this transcription factor, as well as the specific regulatory mechanism it deploys upon early neural induction. And to do so, at our lab, we used an in vitro differentiation model of mouse embryonic stem cells into anterior neural progenitors, which models the earlier stages of mouse neural development, in whose last stage is an heterogeneous mix of the different neural identities that can be found in the most anterior part of the early neural tube, based all on transcriptional profiling. Specifically, our model recapitulates the expression of some midbrain and forebrain markers, such as INGRAIL1 or 6-3 that I am highlighting here in green in this heat map of expression along differentiation. So to reach our objectives, we established 3 representative data collection time points along differentiation, and we obtained this varied set of multi-omic readouts, including bulk and single-cell data sets coming from ZIC2 constitutive knockouts and an endogenously tagged ZIC2 flag HA tagged cell line, which allowed us to obtain ZIC2 genomic binding profiles when performing ChIP-seq. And with all this, I will just focus today on what we observed for day 3 and day 6, which represent the pluripotency exit and transition towards neural induction, or what we call anterior neural progenitors. Since in pluripotency, what we call day 0, our ZIC2 knockout ES cells did not present major morphological, transcriptional, or chromatin defects, which led us to hypothesize that its function might become more relevant at later stages. And this actually connects me to Fragile Nucleosome seminar series, because Istiak Hussain from William Pastor Lab also shared some similar results. And it was really reassuring to see them and confirm that our results match what they observed. So focusing on day 3, what happens at day 3 of differentiation? Well, by differentiation day 3, ZIC2 expression and genomic binding increase. And interestingly, most of the day 3 ZIC2 genomic binding is de novo, which could fit with a relevant role for ZIC2 at this stage. But despite the increase in expression and binding, ZIC2 knockout ES cells differentiated successfully into day 3 progenitors with no evident morphological or transcriptional defects. If so, they were very subtle in comparison to the wild-type controls. But besides their normal appearance, it was the attack sec data which caught our attention. Differential accessibility analysis revealed that our SIG2 knockouts presented a high number of differentially accessible regions at this time point, the majority of them being less accessible sites. And we went further to classify these regions based on their proximity to a TSS, finding that the majority of these regions were distal, meaning that they were found more than 5 kilobases away from a TSS. To resolve whether SIG2 could be directly implicated in regulating the accessibility of these regions, we checked the proportion of the differential accessible regions overlapping a SIG2 peak at day 3. And this analysis uncovered that 75% of the less accessible peaks were actually bound by SIG2 in wild-type conditions, while only 2% of the more accessible peaks of the more accessible regions overlap with SIG2 peaks. This suggested that what SIG2 is doing is promoting the accessibility of specific putative regulatory elements during the transition from pluripotency towards neural fate specification and commitment. And these gains in accessibility that we observed, the more accessible sites, were not directly mediated by SIG2 and reflect indirect or secondary effects mediated by other transcription factors. So we wanted to know what these less accessible regions were involved in. So we did the classical grade functional annotation analysis, and we found that the regions were associated with genes such as LMX1A, LMX1B, and grade 1, and some Wnt signaling ligands. Keep these genes in mind because I'm going to refer to them along the presentation a lot. These genes were responsible for the enrichment of GL terms, like, for instance, midbrain development. All these being processes and functions that become more relevant at later stages of differentiation upon neural specification. So this was telling us that SIG2 seemed to be preparing chromatin for things to happen afterward. We then decided to explore the regions whose accessibility is directly controlled by SIG2 in day 3 cells, but taking also into account SIG2 binding dynamics from day 0. And what we found was that a higher percentage of these distal regions, the ones shown in purple, were not initially bound by SIG2 at day 0, and concomitantly with the binding of SIG2 in wild-type day 3 cells, these regions gain accessibility, as you can see in the attack profiles. And this gain in accessibility was not achieved as efficiently in our knockouts. Moreover, most of these regions were enriched in H3K4 mono in day 3, while only a small fraction was marked with H3K27 acetylation, suggesting that these were preferentially primed and even poised enhancers whose accessibility was being regulated by SIG2 as mouse CSS were exiting pluripotency and transitioning towards neural induction. What we concluded from this block of results was that SIG2 is dispensable for pluripotency exit, but seems to be acting as a pioneer or priming TF, promoting chromatin accessibility at putative neural enhancers. And even though I'm not showing this data today, with a single cell, we found out that this pioneer or priming occurs broadly in the cell population. We move then to the results obtained by day 6 to see how these emerging chromatin changes were affecting later differentiation time points when neural induction already happens. By day 6, SIG2 expression and its xenomic binding slightly decreased with respect to day 3, but still remained at considerable levels. And only a minority of the day 6 SIG2 peaks were de novo. Most SIG2 peaks were already present on day 3 or even on day 0. So the question now was, is this retained binding important at day 6? Differential accessibility analysis at this final time point showed that chromatin accessibility was changing considerably in our SIG2 knockouts, but the proportion of more and less accessible sites was more balanced this time. Nevertheless, SIG2 peaks at day 6 were only significantly enriched in the regions losing accessibility. So what this was indicating us was that SIG2 was still having a direct and predominant role in maintaining or even promoting chromatin accessibility at this final time point too. With all this seen, it was time to see what was happening during neural induction in the absence of SIG2, what is happening to these progenitors. So after confirming that SIG2 was playing an important role promoting accessibility, we wanted to know what was happening transcriptionally. So bulk RNA-seq revealed that by day 6, the number of differentially expressed genes was the highest and was considerably larger than at previous differentiation stages. Detailed examination of the differentially expressed genes revealed that neural ventral markers such as NKX6.1 were upregulated in our knockouts, as well as markers of post-mitotic neurons such as NHLH1 or DCX, which were upregulated in our knockouts. But, however, key midbrain and roof plate markers, being roof plate the most dorsal part of the developing neural tube, were downregulated, such as LMX1A and INRAIL1. And also some Wnt ligands were downregulated, which are also involved in dorsalization, but also in preventing premature neuronal differentiation. So this was the whole picture, and we could also confirm it with our single-cell data set. So having characterized the main defects associated with SIG2 absence in our differentiation protocol, it was time now to really discern direct from indirect effects and to try to find out which were SIG2 direct target genes. For this, I generated the SIG2 degrown cell line, which allowed for conditional degradation of SIG2 at the protein level during the whole differentiation, upon addition of DTAC to the cell culture media. So I differentiate the cells, and whenever I add DTAC13, SIG2 is degraded immediately. So this could mitigate the potential indirect effects or accumulated compensatory mechanisms happening as a consequence of not having SIG2 from the very beginning of the differentiation. So with this cell line, I performed 3 different conditional depletion experiments. First, a total depletion experiment that resembled a constitutive knockout, which helped us to orthogonally validate the main transcriptional defects associated with SIG2 absence during differentiation. Again, we had LMX1A, we had INRAIL1 downregulated, so all the defects were really there again. And then SIG2 was depleted for the last 48 and 24 hours of differentiation, coinciding with the induction of different neural genes. The objective of these 2 experiments was first, again, to reduce the accumulation of possible indirect transcriptional changes, and second, to refine the set of SIG2 direct target genes, focusing specifically on neural induction. We wanted to look for genes that whenever SIG2 is not present, their expression is considerably affected. So one particularly interesting observation in the SIG2 depletion experiments was that while the number of upregulated genes was reduced more than 6-fold, comparing the total depletion to the last 24-hour depletion, the number of downregulated genes remained more constant with only a 2-fold reduction. This indicated that, at least in our model context, and I'm very cautious about this, SIG2 was mainly an activator, and that the upregulated genes we observed in our knockout were most likely indirect and were not mediated by SIG2. Not only we were guessing it because of these dynamics, but also based on what we knew so far on chromatin accessibility data. So guess which genes were downregulated only when only depleting SIG2 for 48 hours or even 24 hours? Well, again, we found LMX1A, LMX1E, INRAIL1, and some Wnt family members. So whenever you don't have SIG2, these genes fail to get induced, representing potential SIG2 direct targets. With all this information, it was time now to narrow down the most relevant SIG2-bound regulatory regions regulating the expression of these targets that we had already identified. And to do so, we leveraged the multi-ohm, single-cell attack, and single-cell RNA-seq coupled, coming from the same cell, data sets that we generated in wild-type and SIG2 knockouts at day 6 anterior neural progenitors, looking for significant correlations between changes in chromatin accessibility at day 6 SIG2 peaks and the expression of nearby genes. So this analysis identified a total of 1,700 links, where the vast majority was like the one I am presenting here for INRAIL1 as an example. These were SIG2 peaks that lost accessibility in our SIG2 knockout cells at day 6, and that were paired with genes, nearby genes, that regulated also in the knockouts. Next, these links were filtered by only keeping those in which the SIG2 peaks overlapped with day 6 active enhancers, meaning they had K27 acetylation, K4 mono, and attack signal. And we only took the genes present among what we call the top direct SIG2 targets that we identified by depleting SIG2 for the last 48 and 24 hours. And we obtained thanks to the degrown cell line. This resulted in a refined set of 71 gene enhancer links containing high confidence SIG2 regulated enhancers and their associated target genes, thus representing a high confidence SIG2-dependent gene regulatory network in anterior neural progenitors. To directly assess the functional relevance of these SIG2-bound enhancers that were part of our SIG2-dependent GRN, we used CRISPR-Cas9 to delete a few of these enhancers present in our final gene enhancer list that were linked to engrade 1. I'm just showing the engrade 1 example, but we also did it for the LMX1A and LMX1B, and we got similar results. We deleted these enhancers in mouse CSS, and we differentiated them to anterior neural progenitors, and we could confirm that the expression of the linked targets was reduced to levels comparable to what was occurring in the SIG2 knockouts. And with this, I use this graphical abstract to conclude that during our in vitro neural differentiation system, we found that even though I didn't enter into this part of the project, SIG2 function seems to be redundant and partially compensated by SIG3, where they are involved in regulating pluripotency genes. Then, upon pluripotency exit, SIG2 is acting as a promiscuous sort of pioneer or bookmarking transcription factor that primes distal enhancers. And upon neural induction, when neural progenitors can already be found, SIG2 selectively activates enhancers regulating midbrain and roof plate patterning genes. This was a small and very simplified part of the story, but if you are curious about the full article, you can check it. It's a preprint. And yeah, we are with the revisions now. So I just wanted to thank, this project is the result of being part of a fantastic and supportive lab led by Alvaro. I want to thank especially Victor, who is the co-author of this article, whose bioinformatics skills have been essential. And also, it comes as a result of a fantastic collaboration with the Q Mignon and Judith Solak labs at EMBL when I was doing my PhD internship in what I would say were the 3 of the best months of my very short scientific career. So it is my first time sharing this story completely finished, and I will be happy to take questions, any comments or suggestions. So thank you very much.
Awesome. Thanks so much, Maria. Anyone in the audience, if you'd like to ask questions, if you want to ask it yourself, you can raise your hand and I can unmute you, or you can type things into the Q&A. While we give everyone time to either come up with their questions or type them in, I'll start by saying so there's 23,000 or so ZIG2 peaks where you don't see changes in accessibility. Do you have any inclination as to what it's doing there? Like, I wonder if maybe it's binding to other regions of the genome might have more of a role, and maybe there it's playing more of a role as a repressor, and that's where you're getting the upregulated genes.
In the regions where it's binding, you mean? So in you are referring to the day 3 or to the
day 3, yeah, where you see that 6,000 peaks, 70 or 6,000 or so peaks overlap with accessibility changes, but you've got another 23,000 that don't. Yeah.
So first of all, I mean, I could answer different ways. Here at day 3, there is still a bit of SIG3 expression. So one could, and we've shown in the project that they bind similar regions. So there could be some sort of compensation there. Into what it's doing there, it might be that it's doing nothing. There are reports that say that some binding is non-functional, like it's been said. So we decided to focus on the ones that were actually showing a functional relevance. This is why we went, to further analyze them. But what it's doing there, we don't know. We could check it functionally too. But since this part of the story was not what we were really interested in, which was the neural induction, we decided to not go deeper there.
Sure. So we have a question from Azim Khan in the Q&A asking about the changes in accessibility at pluripotency markers. Do you see?
Yeah, again, not really. Sorry, again. So if you go to the preprint, there is the two first figures go into SIG2 and SIG3 redundancy, and I mean, to us it seems like in terms of pluripotency genes, the leader is SIG3, or at least both act cooperatively. We don't see much because in our knockout, we still have SIG3. When we've deleted SIG3, there are major changes in pluripotency genes expression. I mean, actually, our SIG3 knockouts differentiate into endoderm, and they cannot retain pluripotency. So we don't see it, probably because we still have SIG3 there, you know? So this is what.
Yeah. And now Sheila asks, great talk, interesting to see the priming effect early in differentiation. If you were to push the differentiation of ZIC2 knockout cells, do you eventually see a defect?
So by pushing it, so I don't think I completely understand the question. So
I don't know if Sheila wants to clarify, or
sorry.
yeah, it's okay. I guess I don't like if you're really like I wonder if you try to drive it down a specific neural lineage, or if you try to pattern them some other way.
Whether we would see some changes there.
Sheila has her hand up, so I'll let her come on and ask the question herself. Let's see, allowed to talk. All right, go ahead, Sheila.
Okay, sorry, I forgot how to ask to unmute. um, I was wondering if, like, during the differentiation, you'll see eventually that cells die or they stop differentiating during, like, with the knockout of ZIC2, like, because it's such a big effect, right?
Yeah, we never brought it like longer than day 6. What I could say is that if we keep extending it, we will eventually have the radial glial cells, and then we have neurons. So we clearly find neurons in our SIG2 knockouts. So I would say if I push it, I would need to add something to maintain fully differentiated neurons. This is something we observed in the single cell data, but also when doing an IF for TUG1, which is this dendrite classical marker, I do see that the SIG2 knockouts, they show neurons. So, and this is strictly connected to what it is doing with the Wnt most likely, because SIG2 is activating some of the Wnt family members, which are indeed dorsalizing, but also they eventually put the brakes on the premature differentiation. So yeah, this is more or less what I would say would happen, but it's true that I haven't done the experiment of bringing them to day 8.
Awesome. Well, we're hitting time here, so if anyone else has further questions for Maria, please go ahead and put them in the Q&A, and she can continue to answer questions as we move into our second talk.